Hello everyone. In today's section, we shall look into the purine and pyrimidine metabolism. There are a number of amino acids that are synthesized into purine and pyrimidine nucleotide. Nucleotides can be recycled from nucleic acid and cofactors that are broken down. And since these purines and pyrimidines can be synthesized in de novo in our body, as I said, from several amino acids, no specific dietary requirement is necessary. And they are the important energy currencies, such as the ATP, and even you have seen GTP. UTP, which is uridine triphosphate, is the source of energy for activation of glucose and galactose. Whereas your CTP, that is your cytidine triphosphate, is the source of lipid metabolism. AMP is a part of coenzymes like NAD and coenzyme CoA. So both your purine and pyrimidine, as said earlier, are part of your nucleic acid. So first we shall look into the pyrimidine metabolism, that is how they are synthesized, and also the breakdown pathway. So if you look into the slide, the cytosine, look at the structure, cytosine, uracil and thymine are your pyrimides. And uracil is an analog to thymine in RNA. Okay, see in the RNA, you do not find thymine. Instead of thymine, we have the uracil. So what are the raw materials that are required for the synthesis of the pyrimidine? We require glutamine, aspartate, tetrahydrofolate, and folic acid, that is your vitamin B9. So here I have shown an example of uridine monophosphate. So whenever there is synthesis of pyrimidine, first the bases are formed. Okay, you know that we have the nitrogen bases, the sugar group, and the phosphate group. In the pyrimidine synthesis, first the bases are formed and these bases are attached on the sugar molecule. And here you can see a process wherein your uridine triphosphate is converted into cytosine triphosphate. So you can see that the UTP in the presence of the enzyme CTP synthesis or synthetase gets converted into CTP, wherein it utilizes the amino acid glutamine and gets converted into glutamate. The amine group of the glutamine has gone and attached into this nitrogen bases, and this forms the cytosine triphosphate. You can just compare the structure of these two here, the bases, and it utilizes a molecule of ATP and also water. So throughout the steps you have to remember, that is, when the UMP, your uridine monophosphate or uridine diphosphate or triphosphate, when they're going from a smaller molecule to the larger molecules, because you have in uridine monophosphate means one phosphate group, and triphosphate means, as you see here, three phosphate group. So what is adding this triphosphate? It is the enzyme kinase. So that is what we have taken one example and we have shown here. So your CTP synthetase, okay, this enzyme makes the CTP from UTP. So you have to remember this one point, important point. Whenever there is a pyrimidine formation, first the basis is formed, and basis means this, and then they are attached to your sugar group. Okay. And another point also you have to keep in the mind is, now, for example, it was uridine and cytosine, what we were speaking. Similarly, you have another group, thymine also. But we will look into the basic synthesis, what is happening in the pyrimidine metabolism. So, to start with, we have what? We have the glutamine, we have two molecules of ATP and carbon dioxide. So these are utilized by your cells. These all are utilized by your cells, and then they are converted to carbamide phosphate. How? By the enzyme carbamide phosphate synthase. So it is an enzymatic process. And also you see that the glutamine is converted to glutamate. That is, the glutamine is giving away its amino group and getting converted to glutamate. And the enzyme carbamide phosphate synthase, which is here, this name you have already heard of. So where did you hear this? You have heard this in the urea cycle. Okay, so the starting enzyme even in your urea cycle, which first occurs in the liver mitochondria, there also you have seen the same enzyme carbamide phosphate synthase 1, but this is 2, which is present in the cytosol. And also the green color, the arrow mark, and the red shows you that whether they are having inhibitory effect. Red is inhibitory effect and the green is the activators. So I'm not going to go into the regulation part, but you can see everywhere that wherever there is regulation required, you can understand by the line. Red line means it is the inhibitor, and if it is a green line, it is an activator of the reaction. So we see here a carbamyl phosphate is produced and glutamate is produced. Next, your carbamyl phosphate then gets converted into carbamyl aspartate in the presence of another enzyme, aspartate transcarbamylase, wherein it requires an addition of aspartate group. So with this, in the presence of the enzyme aspartate transcarbamylase, and in the presence of the enzyme aspartate, we got carbamyl aspartate, and again in the presence of the enzyme dihydroorate dehydrogenase, we get the orotic acid. So kindly remember, the orotic acid is the first nitrogen pyrimidine to be produced. Okay, it is the first nitrogen pyrimidine to be produced. It's not the entire base which is being formed. And then you have your pentose pathway, that is your pentose pathway which is occurring in your body, wherein it produces the ribose 5 phosphate. This is a very important step in your nucleic acid metabolism. Without this, you don't find any synthesis happening, because this is the one which is producing your ribose 5 phosphate. What is this ribose 5 phosphate? It is the sugar molecule, right? So you need a sugar molecule, whether it is an RNA or DNA. because onto this sugar molecules is what your nitrogen bases sit on. And it is to these sugar molecules your phosphate groups are attached to. So pentose phosphate produces ribose phosphate and which gets converted into 5-phosphoribosal 1-pyrophosphate in the presence of the enzyme PRPP synthase. So 5-phosphoribosal 1-pyrophosphate. This in turn combines with the orotic acid and forms the orotidyne 5' monophosphate in it is abbreviated as OMP. So this steps you have to remember and be careful. So orotic acid is formed. That is your first nitrogen is being formed for the pyrimidine. At the same time, what happens, you want to your sugar molecules, and the sugar molecules is being produced by your pentose phosphate pathway. What is your sugar molecules? What you require is the 5-phosphoribosal 1-pyrophosphate, that is PRPP, and they combine to form the orotidyne 5' monophosphate, which is called as OMP. So here you can see that your OMP in the presence of the enzyme OMP decarboxylase is converted to UMP, that is uridine monophosphate, which in turn gets converted to UDP in the presence of the enzyme ribonucleotide reductase, which in turn gets converted to UDP, that is DUDP. D means deoxyuridine diphosphate. Okay, and this DUDP is then converted into DUNP, in turn gets converted to DTNP, that is deoxythymidine monophosphate. Now this step wherein the conversion of deoxy-UMP to deoxy-TMP is very important because it is catalyzed by the enzyme thymidylate synthase. Okay, synthase means always the formation, production, okay, synthesis. And this requires a cofactor. Okay, the cofactor you can see it is N6N10 methylene tetrahydrofolate, which it gets converted into tetrahydrofolate. Now, this in turn, this N6N9 methylene tetrahydrofolate, that is, it is requiring, we require folic acid for this synthesis. So this is an important enzyme because this in turn, no, it is controlled by another regulator which we have not shown here, that is dihydrofolate reductase. So why is this important? Your dihydrofolate reductase means that most of your chemotherapy drug, or that is your cancer drugs and abortion drugs, act on the dihydrofolate reductase. So it acts on the folic acid, thus the thymidine or your thymidinylate synthase is inhibited. So we will not go much into these topics, which okay, we'll just deal with general biochemistry, but a point to remember. So you got your DTMP, that is your deoxythymidine monophosphate, which then converts into deoxy-TDP, and then which gets converted to deoxythymidine triphosphate. And this enters into the DNA synthesis or the formation of the DNA synthesis. In the next, you will see that you have some more reactions here. These are known as your side reactions. So this side reaction, what happens, this is your main reaction, right? The UDP can also get converted to UTP, uridine triphosphate. And in the presence of the enzyme CTP synthase, they get converted into cytosine triphosphate or cytidine triphosphate. So this happens with the addition of ATP and glutamine. We get a cytidine triphosphate in the presence of the enzyme CTP synthase. And these together, your UTP and CTP, form the RNA synthesis in the presence of the enzyme RNA synthase. So you see that your CTP is then converted to CDP and then deoxycytidine diphosphate in the presence of the enzyme ribonucleotide reductase, and then we can see that getting converted to deoxy TTP and then again entering into your DNA synthesis. So both, okay, your direct pathway, that is your deoxy thymidine triphosphate or your deoxycytidine triphosphate, both enter into the DNA synthesis, that is the formation of DNA. Whereas a side reaction, what you're seeing, wherein your UTP and CTP can enter into the RNA production with the help of the enzyme RNA synthase. So that is about, in a simple way, to explain your pyrimidine synthesis, how the being produced. Now let us look into the pyrimidine catabolism. As you all know, catabolism is the breaking down of your metabolic products. So here we have taken what? DNA. So in DNA, what is the nitrogen base present? It is your cytosine and the thymine. The cytosine gets converted to uracil by a process of deaminization where ammonia is released. And both uracil and thymine can enter into pyrimidine salvage pathway. Salvage pathway means the salvage pathway is in which your biological products is produced from intermediates in degradative pathway. So your biological products which are produced by your intermediate degradative pathway of its own substances are known as salvage pathway. Okay, keeping that apart, you can see that your uracil and thymine get converted into dihydrouracil and dihydrothymine by the rate limiting step dihydropyrimidine dehydrogenase. So this is a very important step which controls your overall pyrimidine metabolism. So during this formation of your hydrouracil or hydrothymine, what happens is your hydrogen molecule is being removed. Okay, from where? From your NADPH. And finally, your dihydrouracil forms the N-carbonyl beta alanine and N-carbonyl amino isobutyrate. So next comes another enzyme known as beta-uridopropionase, which can convert your N-carbonyl beta alanine in the presence of water to beta alanine. And here your N-carbonyl amino isobutyrate gets converted into beta amino isobutyrate. And both of them, that is your beta alanine and beta amino isobutyrate, produces a byproduct which is known as ammonia and carbon dioxide. So you all know that ammonia is toxic to your body and it has to be thrown away from the body, which is done with the help of the kidney. And the most important organelle which comes here in for the rescue is the liver mitochondria, which converts this toxic ammonia into the urea through the urea cycle, and it is excreted. And again, what happens to your beta alanine and beta amino isobutyrate? They get converted into the respective acetyl CoA and succinyl CoA. And you are familiar with these two words, you know that they are the intermediate compound of your PCA cycle or Krebs cycle, or otherwise you call it a tricarboxylic acid cycle. So once again, this acetyl-CoA and succinyl-CoA, they enter into your PCA cycle, forming then many intermediate components and producing what? ATP or your GTP. Ultimately, the energy is being produced. So the next we would like to look into the purine synthesis and catabolism. Uh, so whenever we spoke about the nitrogen, the pyrimidine, we said that it is you have what cytosine, you have your uracil and thymine. And these were what were the structure? Look at the structure. There were six-member nitrogenous bases. Okay, the structure is quite simple. Whereas when you come to the purine, you can look at the structure. The purine is a heterocyclic aromatic organic compound that consists of two rings, a six-member nitrogen ring, same as that of in your pyrimidine, and also it has a five-membered imidazole ring which they are fused together to form adenine and thymine. Whether it is your RNA or DNA, it is only the adenine and thymine. So this requires the formation of glutamate, glycine, tetrahydrofolate, and aspartate. So coming to the metabolism, it is a very long reaction here, and we will not be speaking about all the reactions. We will just touch upon the important steps. Here you see the first, what is being produced is a pentose phosphate pathway. Did you see this? Yes, you saw in the pyrimidine production, but it was afterwards, after the orotic acid being formed, pentose phosphate pathway occurred. But here what happens? First the pentose phosphate occurs. That is, first a sugar group is being produced, and on top of it, your nitrogen bases will get attached. But in pyrimidine mode, first your nitrogen base is being produced, and all these nitrogen bases, once they are formed, they go and sit on your ribose sugar. So that's the important point you have to remember. So the pentose phosphate reduces the ribose 5-phosphate, which in turn, in the presence of the enzyme PRPP synthase, it produces 5-phosphoribosal 1 pyrophosphate, and this in turn gets converted into 5-phosphoribosal 1 amino. In the presence of the enzyme glutamine PRPP amido transferase, and this is the first committed step, wherein you can see that your glutamine is converted to glutamate. Sorry for that, you can't read this word. So your glutamine is being converted to, this is glutamate. Okay, so finally my 5-phospho-ribosal 1 amine is being produced, and then you see a series of reactions, or 10 to 14 steps reaction to finally form the inosine 5-monophosphate, your starting material, your hydrogen. Okay, this is the structure of the inosine 5-monophosphate, or it is also called as the IMP. So what it requires? It requires glycine, ATP, N10 formyl, PHF. It requires glutamine, ATP, carbon dioxide. It requires aspartate and ATP and N10 formyl tetrahydrofolate. So these all are involved, finally forming the inosine 5' monophosphate. Look at the structure here. So here what happens? You can see that the nitrogen, okay, it is given by whom? It is being produced by donated by your glutamine. Your nitrogen and your carbon group is produced here by your glycine, the red one you're seeing. So this is your carbon and nitrogen, which is being donated by your glycine. And what is your aspartate? Here, your aspartate produces the amine group, and your carbonyl here, you can see it is produced by your bicarbonate. And we have the CH groups which are produced from the formyl tetrahydrofoliate. So these were the raw materials, right? What we saw initially. And look at this, all the raw materials have joined together along with your pentose phosphate pathway, which produces your phosphoribosyl pyrophosphate, forming the IMP. So next, what happens to this IMP and how the synthesis is being formed, we shall look into this. And similarly, here also, when we saw the structure of this uridine monophosphate, you see here, okay, your amine group, similarly, it is produced, donated by your glutamine. And half of the cycle of this nitrogen base is nothing but your aspartate, right? And again, your carbonyl group is given by the your tetrahydrofolate. So what is my starting material for the production of adenine and guanine nitrogen base? It is your ribose 5-phosphate INP, inosine monophosphate, whatever we saw in the previous slide. Now this is the starting material for both ANP and GMP. You can see here, I'll be just mentioning in short forms, okay? So your INP, it forms what? Ribose 5-phosphate adenylosuccinate. So here what is happening? You can see this entire portion is nothing but your aspartate. That is the entire aspartate. Your entire aspartate molecules goes and binds to this. It is added here. And here your GTP is being required. That is your GTP is being utilized to form this adenylosuccinate. So your ribose 5-phosphate requires aspartate, which donates the complete aspartate is added, and it also requires the GTP. So here it is. And next what happened? Next you see here what is being formed? Amino monophosphate is being produced. Okay. So what happens is that we have, I mean the picture is cut off here. So what happens is we are retaining here only the amine group, right? The amine group of what? From where did I get? I got the amine group from my aspartate, and others are removed. How they are removed? They are removed in the form of fumarate, and I'm left with this structure, and this structure is nothing but your ANP, adenosine monophosphate. That is, you have a ribose 5-phosphate, that sugar group. So you can see how the change happens in this carbonyl group, wherein your aspartate attaches and it retains the amine group. Next, coming to the formation of GMP. To form GMP, what happens? Here you can see here, I'm very sorry for the slides keeping jumping. Here you can see that the water molecule is oxidized. So, and then the carbamide, and then again they are replaced by another carbamide group with the amino group from the glutamine, and this step requires ATP. So in the first step, what happens? You have your NAD and your water. That is, your water is oxidized to a carbamide, and then it is replaced by the amino group. And That is, it is again replaced by a carbonyl with an amine group from the from where from the glutamine, and it requires ATP. So these are the two important steps you have to remember. And you can see here the intermediate wherein your water is getting oxidized. You can see what is being formed: ribose 5 phosphate and xanthosin monophosphate is being produced. Okay, so this is the oxidized your water molecule wherein it is getting oxidized, and in the next step you see your that carbonyl, and then again this carbonyl being replaced, okay, by the amino group. Here it is replaced by the amino group. So your carbonyl group which is being produced by the water is then replaced by the amino group. From where it gets this amino group, it gets it from the glutamine. And what is being utilized? ATP is utilized, and finally the glutamine, after donating its amino group, forms the glutamate and then AMP and inorganic pyrophosphate. Thus your GMP is produced. So you can see that from IMP you're getting AMP and GMP by a two-step reaction. So you can remember this easily. For the formation of adenosine monophosphate, what I require is A and A, that is aspartate and adenosine monophosphate. Whereas you can see that G, what is most important is the glutamine, because it is my glutamine which is providing the amino group from the glutamine, and it also, I told you, requires ATP, and then it gets converted into the glutamate and AMP. And here the water molecule is very important, where the water oxidizes your carbonyl group, and your carbonyl group is again replaced by the amino group. Now coming to the purine catabolism, you can see it is a quite complex, but it is a very simple reaction. We start off with the AMP. This AMP can be converted into IMP by the enzyme, or it is processed to IMP in the presence of the enzyme AMP deaminase, wherein an amino group is released. AMP can also be processed to adenosine in the presence of adenosine deaminase ADA, and adenosine can get processed to inosine. So your AMP can be processed to IMP or into adenosine, and adenosine in turn can be processed to inosine. Both you can see that they give away their ammonia group. And this enzyme adenosine deaminase is a very interesting group of enzyme, because this any deficiency in this enzyme causes the severe combined immune disease. Okay, SCID, the SCID disease, severe combined immune disorders. These are, which are very high, difficult. At least 15% of the population has this type of disease, and it is leads to ultimately death of the individuals. So we have ended up in inosine. This inosine, okay, it can get processed to hypoxanthine in the presence of the enzyme purine nucleoside phosphorylase. Purine nucleotide phosphorylase by the removal of ribose 1 phosphate. So what did I get? From inosine I got hypoxanthine. This hypoxanthine can enter into the salvage pathway. Otherwise what it can do is it can get converted. Hypoxanthine can be getting converted into xanthine by the formation of hydrogen peroxide. And this xanthine can then again get converted to uric acid. This uric acid has to be removed from your body into the blood, and then finally it has to be excreted in the kidney, because this uric acid can cause a accumulation of uric acid can result in a disease known as gout, GOUT. And this enzyme, okay, for the conversion of hypoxanthine to xanthine, xanthine to uric acid, this we have an enzyme known as xanthine oxidase. So it is catalyzed by the xanthine oxidase. So we have to regulate this enzyme xanthine oxidase, and drugs are being produced for this xanthine oxidase so that we see to that there is no accumulation of uric acid, and we can inhibit or prohibit the disease which is known as gout, GOUT. Okay, so you can see that how finally they end up in xanthine and forming uric acid, and they get excreted from our body. What about your GMP? Till now we spoke about AMP. What about your GMP? The GMP can get converted to guanosine wherever you can see that arrow marks. Okay, they are reversible reactions. So your guanosine can get converted into guanine by your purine nucleoside phosphorylase. Okay, same purine nucleoside phosphorylase. So with the removal of ribose 1 phosphate, it forms guanine. This guanine can enter into the salvage pathway. Otherwise, in the presence of the enzyme guanosinase, okay, NASE, it is, I don't think so they are legible. Okay, ASC, this is okay, guanosinase, and ammonia is being released, and then it gets converted back to xanthine, wherein the xanthine again produces hydrogen peroxide and form the uric acid. So you can see that both your AMP and your GMP both can form the xanthine, and you can see that the hydrogen peroxide is being produced, and ultimately your xanthine gets converted into uric acid. Otherwise, the both of them can enter into the salvage pathway. If they enter into the salvage pathway, what happens is your hypoxanthine and guanine, okay, they get your hypoxanthine in turn gets converted to IMP. So once it gets converted into IMP, you know that you can always synthesize your AMP and GMP, right? So your guanine can get converted into GMP and your hypoxanthine into IMP. It is catalyzed by the reaction hypoxanthine guanine phosphoribosal transferase, wherein your phosphoribosal pyrophosphate is being converted. I mean, a pyrophosphate group is being released and finally getting converted to IMP and GMP. The IMP can further be processed to AMP, and AMP and GMP, which are formed through your salvage pathway, purine salvage pathway, can again get into the synthesis process. With that, we come to the end of the class of your purine and pyrimidine metabolism. Also, you saw that in some of the cycles we did not go fully into it because of the food. I told you particularly in the purine synthesis, because the length of the reaction is quite big. You can write it in the similar form, not an issue. Okay. So we saw how the pyrimidine and purine are synthesized and how they are being catalyzed. Sorry, broken down by the enzymatic reactions, which catalyzes all these reactions. Hello everyone. In this section, we shall look into the details of biosynthesis of purine nucleotides. The ribose 5-phosphate is produced in the HMP pathway, or pentose phosphate pathway is the starting material for purine nucleotide synthesis. It reacts with ATP to form 5-phosphoribosal 1-pyrophosphate in the presence of the enzyme ribose phosphate pyrophosphokinases. In the second step, the glutamine transfers its amide nitrogen to the PRPP to replace the pyrophosphate and produce 5-phosphoribosylamine. This reaction is a committed step in the purine synthesis. Next, you can see that the phosphoribosylamine reacts with the glycine in the presence of ATP to form glycine amide ribosyl phosphate, which is catalyzed by the enzyme synthase. This is followed by a formyl group is added and formyl glycine amide ribosyl 5-phosphate is formed in the presence of the enzyme formyl transferase. This is followed by another reaction wherein the glutamine transfers the second amino acid group to produce 5-amino imidosyl ribosyl 5-phosphate. So we have utilized two glutamines, that is in this step, and this is the second step. This is followed by incorporation of carbon dioxide. The reaction is carboxylation occurs to yield 5-amino imidosyl carboxylase ribosyl 5-phosphate. The next reaction occurs in the presence of the amino acid aspartate, where ATP is utilized in the presence of the enzyme synthase to produce 5-amino imidosyl 4-ribosyl 5-phosphate. In the next reaction, you will see that the formation of 5-amino imidosyl 4-carboxyamide ribose phosphate, which occurs in the presence of the enzyme adenylosuccinate lyase, releasing the fumarate. That is, the aspartate donates its amine group and gets converted into fumarate, releasing the fumarate. And this is followed by another reaction wherein a formyl group is added to the amino group to produce 5-formyl amino imidosyl 4-carboxamide ribosyl 5-phosphate. In the next step, you can see the ring closure occurs to form inosine monophosphate or inosine acid with an elimination of water in the presence of the enzyme cyclohydrolase. So this is an important reaction because this is where the cyclation of the imidosyl group occurs. This is followed by the production of imidosyl monophosphate, and this imidosyl monophosphate can then enter into the reaction to produce AMP and GMP. So this is the whole reaction which speaks about the details of the purine synthesis or the metabolism. So you have the important steps, we can just recap it. That is the ribulose 5-phosphate, then we have the glutamine, then the glycine, then the formyl group, which is followed by glutamine, and then the carbon dioxide. We have the aspartate, and then we have again the formyl group, and then the water is removed from here, and then the oxidization steps occur. So these are the steps which are involved in the formation of IMP or the inosine monophosphate, and this is the starting material for the production of adenosine monophosphate and guanine monophosphate, which we have already dealt with.